So I guess I'm scheduled to be the next speaker. So the topic that I'm going to be talking about is updating where we are with this pH upregulation and as an adaptive capability, if you like, or a buffering that it may give corals to the, I think, the inevitable issues of pH, of ocean acidification. So this work, I'll briefly show some of Michael's, just to reiterate some of Michael Holcomb's work. And it's been involved Jim, who just spoke earlier, and Julie Trotter did some of the early work on this particularly. Okay, so ocean acidification. This is well known, I think, to most people in the audience. And basically, we're putting a spike of CO2 in the atmosphere. This goes back to 10,000 years. It's unprecedented. We're above 400. We just hit 400, I think, in Hawaii in May this year. My view, this trend is going to keep on going. And the surface oceans are in equilibrium with this increasing atmospheric CO2. And for acidification, what's really happening, the key thing is that the carbonate ion curve is shifting to the left as pH declines. And the key reaction, we believe, is that carbonate plus calcium is driving calcification. So the first order, then, pH is likely a major potential threat to calcification. But we really have to understand, I think, what's happening with the calcification process. And Michael showed yesterday quite nicely that although the seawater is changing its composition, it's really at the interface or the calcifying fluid where the calcification is occurring. That's the key process. And it's been well known, in fact, that corals do calcify at a different pH. So they have means to increase the pH. And by doing so, they increase the saturation state, the carbonate ion concentration. And they do this by pumping calcium into the calcification site in exchange for hydrogen ions. So that's one way. This is the major way that they internally modulate their pH. And the basic point of this story if you don't remember anything else, is that this pH at this calcifying region here, we call it pH calcifying fluid, has this relationship, roughly a half to a third of that of the seawater pH here, with an offset that's species dependent. And this seems to be a very robust, amazingly systematic relationship that I'll be showing you. Now, the other point to make is that although, of course, this is Ken Caldera's diagram, which many would know, we're obviously acidifying surface waters. This will penetrate into the deep ocean, so it's going to affect deep sea corals as well. And the timescales of this process has now been set in train. We're with it for several thousand years, or thousands of years. It's inevitable. The only issue is what is going to be the final change in, how much are we going to do it, or what's the extent of it. But I missed explaining one thing, was that this is the early calcification data that Chris Langdon and Atkinson published, the response of calcification to aragonite saturation state, and which is most people would refer to. And you can see that there's quite a lot of dispersion in this data. And if you read that paper carefully, those authors were quite careful in describing what they thought was certain and what was uncertain. But that's mostly been overlooked. Mainly these linear time of responses have been emphasised. And what I'm now showing is, on the right here, is a compilation that John Pendolfi did in 2011. And you'll see that rather than kind of clarifying the situation, it's actually, I'd say, confused it more. So this is a case, more data, more confusion, right? So that, we've got to understand the processes, right, to try and interpret this array of data. I mean, they actually did put omega saturation state -1 on this axis and this line here, and they were plotting seawater aragonite saturation. The simple transformation you have to make is think of the sensitivity depends on the saturation state of the calcifying fluid. And in fact, I'll show you that calcification can occur when you get under saturated waters. And so it's well known now that these kind of diagrams of how the saturation state is going to progress with time have been done by numbers of authors. And this is a recent one from 2013, just published. And the interesting thing, these authors have looked carefully at the changing aragonite saturation state of coral reefs, of reef waters, at least waters adjacent to reefs. Now, as Jim Folter just showed you, in fact, that equals reef waters over any kind of reasonable timescale. So we know pretty much then the likely trajectory of reef waters. What we don't know is the calcification response. Right, whoops. We're doing jumps, I think. Okay, sorry. So as Michael mentioned yesterday, I'll just very briefly go over it. We have another system. By pure chance, nature has provided us with a complement to the carbonate system. It's the borate system. It's actually quite abundant in seawater, and it actually has similar pH relationships as the carbonate bicarbonate system, almost a mirror, almost a dead copy, if you like. And so the amount of borate in the oceans depends on the pH, and this is a red curve. And it turns out that carbonates or corals take up this borate species, the red one, and also by pure coincidence, well, there's a fractionation factor between these two boric and borate species of over 27 per mil, which is given in this, that's parts per thousand. And this is something we can measure with now very, very high precision. We can measure better than a few tenths per mil. So we now have a fine scale pH meter, and it's actually registering the pH of the calcification product. So we're looking at the pH of the calcifying fluid, right? And what I'll talk about is that ideally, if the system was ideal, all corals would follow. If they just took up their carbonate and had the same pH as seawater, they would lie on this curve. But what we find is that if you plot the seawater pH, they're always elevated. And so the difference between what we measure here and what the seawater pH is what we call the delta pH. So that's the additional bit of pH that the corals give themselves to enable calcification to proceed, and it's driving that things up to higher saturation state. So this then is the results from the boron work, which we've now published. This was published last year. And this shows the delta pH, this increment of pH that the corals do as a function of changing seawater pH. And these are numbers of different species. You'll see Acropora, Parietes, Stylophora, and this red one's actually a temperate Mediterranean coral. And you see remarkably that they form these linear arrays, which tells you that the pH upregulation is very, very systematic, telling you that the calcium ATPase pumps are working as very fine pH modulators. But interesting, I'll talk a bit more about, not all things upregulate, a lot of forams don't. And so this is the relationship that we've been able to take, and I want to show you a bit more details about it in the coming slides. Oh, okay, sorry, I'd forgotten. Of course, there had been some measurements of the internal pH. People had put, this paper, they'd put a pH probe into the calcifying fluid, if you can believe that, and measured that increment in pH. So this is a direct measurement using a micro pH probe. And then Michael showed yesterday the use of the dyes, and I'll expand on that, where they've measured the pH using these pH-sensitive dyes. So this is a state just a year or so back. And so it looked like the first order, things looked good with the conventional data, but we didn't know, there was no other confirmation of the slopes that we'd derived from our boron work. What I'm going to do now is we've now looked at a few other calcifiers, and this is some work we've done off the Tasmanian seamounts. And so this shows you the pH of that region of the ocean. If you go down to 3,000 metres, you'll see that the pH, it's the saturation state plotted here, is less than 1, right? So this is a function of changing pH. So ash is the aragonite saturation horizon. So this is where aragonite will dissolve itself because it's insoluble at that point. So so we would not expect any corals normally to exist below this aragonite saturation horizon. Calcite has a deeper saturation. You need to go to a lower pH for it to become unstable and dissolve itself. So, but actually when you look at the aragonitic corals available from this kind of sampling, this you have to use deep sea ROVs and so on to do it. Interestingly, there are many deep sea corals that exist below the aragonite saturation state, i.e. they're growing in saturation state waters less than 1. So that points to this pH upregulation. And so what the story is that I just showed you all this data here for the regular corals that I described, our tropical corals with zooxanthellae and such, etc. And this is the array now that we have for deep sea corals, and these are all the different species. The red one is this very nice picture of this Desmophyllum dianthus. So that's existing down to over 1,000 metres. It's making very nice aragonite septa, and it seems to have a subparallel relationship to the surface coral. So this phenomena of pH upregulation is, and these, although, and these are they don't have any zooxanthellae in these deep sea corals because they're too deep, there's no light. So this phenomena of pH upregulation seems to be a kind of long-lived trait that they've had to make their aragonite skeletons very efficiently, and it's enabled the deep sea corals to exist below the aragonite saturation horizon. So that's kind of interesting. I'll just show another way to predict it. This is the same data, the seawater pH and delta pH. And actually, we've measured a whole bunch of other deep sea corals. These are calcitic. And you notice there's some suggestion now that they certainly have a much lower elevation in their pH. Some don't elevate at all. So these are the non-regulators. This is just a diagram where I plot, instead of the delta pH, I'm plotting directly the pH of the calcifying fluid. And this is the one-to-one line. So if they lay on this line, there's no regulation. I've shown a little diagram here. So some of these are bamboo corals made of calcite. And we're actually using these to get the long-term trajectory of the deep waters. But these guys, because they're calcite, they probably don't need to upregulate as much, because remember I showed you that the calcite saturation horizon is very much deeper than for aragonite. So therefore, although they're living very close to that under saturation point, they can exist much deeper because they're precipitating calcite rather than aragonite. And just to show you the same problems exist for any managers, this is a brittle star gnawing on one of these bamboo corals, so there's still all these interactive effects with biology. So you can't escape starfish issues. Michael showed briefly yesterday the confocal microscopy, so I won't go into that in too much detail, but this is where this is actually a centimetre high coral of Stylophora. It's been grown least conditioned for 12 months under different pH, and then put into a small chamber where they viewed the tissue or the pH of the fluid right at this point here. And we've gone and measured different parts of it using our BORON system. So we've now got a chance to do a beautiful comparison using two totally different methods to get at the pH. And the results are here. So I'll just show you first, these are the BORON measurements we've done. So we've measured them on this outer edge here, and then we've measured at the apical part, which is really the main growing part. I won't. So this is what is most actively growing. As Michael mentioned, a lot of people tend to work on this lip, this fringe here, because you know it's new growth. But anyway, let's look at the BORON data for that. Sorry, I've jumped a bit. So here's the BORON data for the top part, the solid, and when we look at this part here, we get a very similar array, slightly lower, but almost subparallel. Now, it turned out another lab, this isn't the first time this species has been measured. It had actually been cultured at a lat, and other people had measured the boron on a totally different specimen in a lat, grown for 12 months, and there's their data shown in this orange curve. So two totally different sets of experiments reproduce, at least the boron, on the apical growth, I should emphasise, on that major growth part. And then if you look at the confocal microscopy results that Michael showed yesterday, you'll see that there's almost subparallel, not quite. There's slight differences in the slope, but they're much more depressed, okay? So they have a lower total offset. And he gave reasons for that, and we think it's due to the fact that the tissue is very, very thin on that outer part, so the permeability probably has changed. So the efficiency, or the pumps cannot keep up with, there's probably a lot more leakage of seawater into the calcifying site, and the calcium ATPase pump can't just keep up with it. Although, interestingly, the slopes are very, very similar, so the gradient is maintained. So once we know the gradient, and we know the relative change in pH of the calcifying fluid as PCO changes in the atmosphere. Now, pH is only one of the carbonate parameters, right? So we've got that of the calcifying fluid. We don't actually know the what's called the DIC, the total inorganic carbon. If we just assume it's either constant, and we've done this calculation, it's just 2 times seawater in the calcifying fluid, then the following calculations follow. If you make some other assumption, if you make 1, then you'd probably halve these curves. But what I'll show you, we just have to assume this is constant, right? And I'll present another argument. We think 2 is not a bad number for a total independent line of evidence. So we can now so what I'm showing here then is the saturation state of the calcifying fluid and how it evolves as PCO2 increases in the oceans. So you can see it's not, and this is the evolution that seawater follows, right? So it's now at 400 ppm, so we're less than around 3.5. And of course, the ocean saturation state's going to get down to 2, as I showed you on that other diagram. But the internal saturation states are staying quite high. Although at the edge, that Michael's results for the for that lateral growth at the very edge, you can see the saturation state's much lower because of that increased permeability. So and each of these different colours are different species, right? So we've got all this figured out for different species, and that very flat line there is the latest data from Stylophora pistillata that Michael measured from the Monaco group. And there's an equation you can use then. So the actual calcification rate, G, this is just for abiotic calcification. It's actually well known from especially the geochemical literature, is omega -1 of the calcifying fluid, right, remember, and some other constants. I'll talk to these these are temperature dependent. So we can then predict the calcifying rate knowing the omega of the calcifying fluid. And we're just comparing relative changes. So if you assume you started off here just over 1, these are trajectories that our model predicts. Actually, this is the one we just Jim ran it for the Stylophora from Monaco. You see that we have the very flat trajectory, right? And the general model is here with all the other species, right, that we have. So we've given it this nomenclature, internal pH abiotic calcification. i.e., what we're saying is once the corals manipulate that bit of internal chemistry, from then on the process can more or less be considered as abiotic to first order. But the point is that the non-regulators, this is their trajectories, right? The calcite and all those other things that don't upregulate the pH, they're really strongly affected. That's the blue curve. So that's the other side of the equation. Good news for corals or ragonitic corals, but a lot of other calcifiers, especially blue water, forams and so on, they're in deep trouble. Now, just to show you, there's a paper published just a few weeks ago in PNAS, and this is some corals in the Mexican Gulf, and they measured the change in calcification. And these corals were sitting over hydrothermal springs, so this change in saturation state was due to differences in how much hydrothermal spring water was there. And these have been sitting there for forever, right? So there's no issue of this acclimatization or dipping your coral under extreme pH for a few weeks. And then we ran our model for it, and there's the model, right? It just predicts it perfectly. The orange curve fits this data, and that's using ×2 DIC enrichment. And the other data points are shown here, and so this group we believe is our model. We can now predict calcification as a function of pH or seawater saturation state with very high fidelity and very high confidence. Right. Being reminded that I'll now come to wrapping up. And actually, we can look at relative changes in the rate of relative to present day, and this is shown here. And again, I emphasise that the non-regulators are in for a tough time. So at 1,000 ppm, you're talking still not trivial, but you're talking about perhaps 30% decline in calcification. The other thing is those constants. They're temperature dependent, and as most people realise, in the summertime most things calcify faster. So if you do put a moderate temperature increment in, you can actually boost the calcification. But this is a abiotic kind of calculation, which I actually think is probably irrelevant, because temperature has all these other physiological effects, in particular on bleaching. Once things bleach, there's no calcification, obviously. And this is the part I think we need to do more work on. So the effect of the pH upregulation as temperature changes and the physiological functioning robustness or the fitness of the coral, as it's being affected physiologically, how does that impact on this pH upregulation process? We don't have information on. That's an area we want to work on. So just to wrap up, they do upregulate pH, and we think it's these calcium ATPase proton pumps. It provides a buffering mechanism against acidification. And deep sea corals, not unsurprisingly, they live in the lowest saturation water, do this the most, and they still follow the same rules. At the lateral edge, that work that Michael's done is that there's a difference between apical growth and growth on edges, and this may have some other issues. So we feel we can quantify out to the 21st century, no problem, this taxon-specific level for normal functioning coral, what their calcification will be. We can deconvolve it from other things. But it's limited, the thermal stress, bleaching, all these factors are key in what I call the physiological functioning. So although, impacts from ocean acidification and global warming are clearly inevitable, the magnitude maybe depends on what we do, there's a glimmer of hope for corals if we act now, in particular if we can maintain their normal functioning, or physiological functioning, then this pH upregulation may help combat some of the worst aspects of acidification. So I'll finish at that point. Thanks very much.